So, briefly, I want to talk about an idea I have for how we could really put investments into a futuristic technology that's already being used in many ways, but we need more development and to put everywhere to really help against fight a bunch of viral diseases and more in the next pandemic too. But briefly, just some nomenclature and ideas of how viruses work. Well, in your body has DNA in every cell. DNA is a very stable molecule, and that's why it is used to store genetic information. It can last thousands of years under the right conditions, sometimes even millions of years under the right conditions. But DNA then makes something called RNA. They have a slightly different structure, they're very similar, but RNA is a lot less stable. Then RNA makes proteins, and these proteins make everything in the organism, almost everything else. Not everything, there's always other stuff, but they help make up a lot of the structures. They help make up everything from your fingernails to your nails to structures that hold your skin together. They make enzymes which carry out all the chemical reactions in your body, from carrying oxygen to your immune responses to digestion and all these other things. The reason why we don't use RNA, why we have the intermediate RNA, we don't fully understand, but one reason to have an intermediate RNA is less stable than DNA, but it protects the DNA in some ways because DNA then doesn't have to do all the stuff that protein does or has to directly code for proteins, so it can be a little more kept in like a safe place. Kind of like if you were to work at the library, not taking out the main master copy of books, but you take notes on the books and just take those notes out of the library so you don't damage the book as much, and those notes go into the more dangerous zone where they can get damaged or water can spill on them, but you still have the master copy that is safe. Another reason why RNA can't do as much stuff as proteins can, but it's more easily made by DNA directly. RNA can also can be modified more than DNA, so that lets you have more control. It's like if you copy notes, you can scribble out part of your notes, or you can do other things, notes delete sections of your notes. So one RNA molecule can actually code for different proteins depending on how it's scribbled or rearranged or read differently, and other things like that. But RNA can't nearly do all the stuff that proteins can do structure-wise. So how do viruses work? Well, there's two types of viruses. The first type is RNA viruses. RNA viruses are different than most biological organisms in that they store their genetic information as RNA, not DNA. They're the exception. And then they take this RNA and they somehow turn it back into DNA, and that's complicated, and it's very individualistic, depending on the virus, and insert it into your DNA, and your DNA has now this DNA code by the RNA, which then makes more RNA, which makes the proteins and structure enzyme, and that DNA also makes RNA, which gets from the virus RNA gets reinjected. This RNA is the reason why viruses can store their genetic information as RNA in the organisms, because they're a lot simpler and they replicate so much more. So that means that mutation is not as big deal, and they don't also have to have all this complex machinery. So viruses, one cell might make millions of copies of viruses. So if 20% of the viruses do not work, that's okay. And like in an organism, like in a human being, if 20% of human beings' babies just die, that wouldn't work. But humans have more cellular, so imagine if 20% of your cells did not work, you would not survive even in the womb. You have a majority of your cells working to survive, a vast majority. They can, same as with bacteria and stuff, and also the more pieces you have, the more things can go wrong. A virus can only have like 12 or so genes, but that need to work. So there is a high chance that one of them can get wrong, but you need to have millions of genes that are working for you to stay alive. And if one of them has a big mutation, then you could die, or you would have a serious health consequence. Even the simplest bacteria have thousands of genes. And then RNA also has less sophisticated machinery that themselves, the proteins, can have more mutations and survive more easily. So all this adds up to viruses can afford this. And the thing about RNA viruses, though, is that they tend to be harder to control. And the reason why they tend to be harder to control is because they mutate so much more, which means that, yes, that's a disadvantage, but it also means they can evolve to gain immunity faster, evolve resistance to drugs faster, treatments. It can be hard for your body to fight them. This is, of course, a big generalization, but RNA viruses that are hard to control include, for example, coronaviruses, I believe. I have to double check that. I'll write, if I don't put a comment in the comment section, I was right. HIV, and a bunch of other ones. DNA viruses work more conventionally. Is they make DNA, and they store the genetic information as DNA, which then gets directly injected into your DNA, and that makes RNA and proteins like the regular things. And sometimes they make RNA in the middle that gets converted back into the DNA for a virus. Sometimes the DNA is cut out of is more directly made in the cell. So that's a brief overview of that stuff. Now here is my point: is we're starting to invent machines that can look at a broad range of RNA and DNA in the bodily fluids, from blood, saliva, mucus to urine. And they've also been using water. Now, how these work is something called, are new technologies where we can either amplify a very broad range of DNA, RNA until we can sequence it, or we can use nanopore sequencing technology, which is this really cool idea where we put DNA and RNA, which are like strings, through this tiny little hole. We pull it through, and then we put electricity through it, or light waves, or something else that bonds to it, and different bases interact with the pore differently, and also different bases that make up the RNA DNA may have different electrical conductivity, so we can get a signal. If it's A, it might be more resistive, or T to electricity. Or we could have it that depending on what letter goes through the little pore, different light signals are given off, and we can use that to sequence. It's a lot less accurate than right now than the more expensive genetic sequencing technology, but it's a lot faster and cheaper. And it's out there also. The other one is that we can make a chip with a bunch of different DNA and RNA patterns on it, or something more exotic, but it acts like DNA and RNA, but they have different stuff. There's a bunch of groups of something NA. So we have DNA and RNA, but there's also PNA, SNA, and stuff like that. And they all just have a different backbone, or what's not the middle stuff, but what on the outside holds it together is different. And these things can, if you have a bunch of them on a chip, they can bind to different sequences in RNA and DNA molecules, and you can read out how they bind. And that can let you sequence a variety of things, because you're not looking for one specific sequence. You have a million of them. If you have a lot of small readings, you can add them up to make a big reading. So you read an AT, then you read there's also a GC section. You put them together as an AT-GC section. Figuring out the right order, that's complicated and hard. And we've already developed this technology in the lab to do quick gene sequencing. We've used it in the wild to sequence a bunch of genes in a water sample or fluid sample to look at a bunch of organisms. And we've used it in other settings. So technology exists, it's already been used. And genetic technology is decreasing on order super fast in price, like thousands of times cheaper every year, literally. So I think this technology would be ready soon, especially if you put a massive investment. And I don't think you have to worry about it sequencing as much your DNA, because your DNA is stuck in the cell. There's a lot less of it floating around in the fluids. And your DNA is so much your DNA is so much bigger that these devices are really meant to work with small DNA or RNA molecules, not large ones, because large ones get tangled too much. There's too much stuff in it. It's like trying to feed into a small hole a big giant string of yarn is all tangled, when these holes are meant, instead of just a small, like 1 centimeter or something like that, your DNA is millions of times bigger, easily. So what would be the advantage of this technology? Well, first of all, this technology, if we use it in the next pandemic, track diseases, we wouldn't have to make a test for every single virus or some other diseases. We would just have one test that could detect every virus. This means before the pandemic, it would already be out there. And therefore, we don't have to scale up production or anything. It's just out there. It's ready to go whenever we need it. And we can already be using it to detect other diseases such as HIV, rabies, or cold viruses and stuff like that. And that means that when it starts hitting, we already have tests to show when it starts hitting. A lot faster response, which leads to earlier containment, which leads to it spreading out less. And it also means we can keep places open longer that do not have the virus, so we can be more safe containing it. Also open up, and earlier containment also means that less likely to spread. Another advantage is that we can detect fast mutating regions and stable regions. So some parts of the viruses mutate faster than other parts because they're more. Some parts can change more without killing the virus. Other parts need to be more stable. In the human body, your heart can't mutate that much, or you would die. Your other sensory organs, but you can mutate a lot your skin color, because that's not going to probably kill you right away. Hence the diversity in skin, hair color, and eye color, not in your heart. Now, why do we want to identify regions that are more critical? For virus drugs and vaccine development. That's simple. We can also identify how the virus in a community spreads much better. This can be looking at how, if we identify a strain with a new mutation, you can try to pinpoint how it's spreading, and that gives you a lot of data, because every time a new one pops up, you know what region it came from, and you know it's spreading along specific lines, or any information instead of a test that can't identify between strains. You can also just having early on data and more testing, because people can be testing not only for this virus but for other viruses. You get more information to identify the spread, and also early on it's easier to figure out how it's spreading than later on, because later on you have if everyone's sick, you can't tell who gave it to you. If one person's sick, then you see two people sick, you can narrow it down to one person. An oversimplified case, but the early on, with less people sick, the easier to identify how the virus is spreading, which also leads to better containment, better ideas about how virus spread, so you know better prevention. Is it airborne? Is it waterborne? Is it borne by mosquitoes? You can also identify that also might be drug targets, so if it's a respiratory, it helps figure out if it's respiratory or not, a bunch of things. And you can also even identify people that spread the virus more, so you know who to really target or not. Now also you can identify societal patterns, interactions, which might spread the virus more in the specific cases. But beyond that, if you have better data from a bunch of viruses and diseases, how they spread, you can build a better model for a future virus they don't know, because you can make better guesses. If you know that, cold viruses are mostly spread in this way by and follow this pattern, blah, 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 then a future cold virus, you could kind of make those guesses based on previous cold viruses or assumptions. You can start trying to figure out more models and make those models better. And figure out stuff you might not, like, are teenagers that being spread or are older adults? Sometimes there's surprising stuff in epidemiology, like sometimes it's middle-aged people that get, this is totally different, but sometimes for a lot of things, it's the middle-aged people that get poisoned the most, not young kids or old adults, because young kids don't have access to those same chemicals, and old adults are less likely to use it. But it's a middle one, and that actually happened with Tide Pods. At Tide Pods, there was a peak at a much older age than you would expect, because the really young kids did not actually have access to Tide Pods, and all the warnings were aimed at teenagers when it's really middle-aged people. Okay, so we get all those benefits, that it's going to be already out there, that we're going to be just doing more testing because of other things. We're also going to learn more how it interacts with comorbidities better, because you're going to have much more data. And you might see that having the cold virus and this virus at the same time might be worse. There's some evidence that actually sometimes comorbidities are weird, such as HIV, if it's well treated, might actually give you a slightly higher chance of surviving coronavirus because it tampers down your immune system so it doesn't overreact. That data was a little sketchy, but who knows? It also means there's more incentive to make this whole thing better and invest in it, because if you're making this better, it's not for just one virus, it will be for a bunch of viruses out there and a bunch of ideas. And the same was with investment. And the money saved by this cheaper plan could go elsewhere to actually developing vaccines or other ideas. And finally, all the data from all the diseases, not only how it spreads, could lead to better development of vaccines and stuff as we get better treating stuff. Well, thank you very much for listening. I hope you found this interesting. Please like and subscribe. I'm trying to reach 1,000 subscribers in a year, which is really, really hard. But with your help, I think I can do it. Also, if you have any questions or ideas for future videos or anything, please leave them in the comment section below. Thank you very much. Goodbye.